Hello and welcome to today ASBMB Breakthrough Webinar, Learning from Nature's Switches to Develop New Drug Targets and Biotech Tools. I'm Isabel Casas. I'm the Senior Director of Scientific Publishing and Programs at the ASBMB, and I'm thrilled to be your moderator today. This webinar is the kickoff of our Breakthrough series. We are doing our second year, and today's session is brought to you by the Journal of Biological Chemistry. Before we begin, a few quick reminders. Please submit your questions at the Q&A feature that we have on the webinar page, and we'll address them during the Q&A session at the end of the webinar. The webinar is being recorded, and it will be available on demand on our website. Now, it's my pleasure to introduce today's speakers, Dr. Kevin Gardner. Kevin is the founding director of the Structural Biology Institute at the CUNY Advanced Science Research Center and a distinguished Einstein Professor of Chemistry and Biochemistry at the City College of New York. His research combines approaches ranging from biochemistry to drug discovery to uncover atomic-level mechanisms that cells use to sense and respond to changes in their environment. This work has advanced our understanding of fundamental biology and enabled new strategies to control protein activity, including applications in cancer therapeutics and optogenetic tools widely used in academia and biotech. Kevin is also an editorial board member of the JBC and the chair of the ASBMB Public Affairs Advisory Committee. Without further ado, please join me in welcoming Dr. Gardner. Kevin, thank you so much, and the floor is yours.
Thank you, Isabel, for the very gracious introduction. Thanks to all of you who are either online or here in a room in New York. Glad to have you all together and have the privilege of being able to share a little bit of our research that we've been doing in my groups, either here in New York City, as shown on the slides or on the picture of our neighborhood, or elsewhere, including Dallas. Confirming that slides are coming through okay, Isabel, is that correct? Wonderful, thanks. So with that, let me start acknowledgements off. It is an April 15th, so thanks to all of you on the call today who have already paid or will be paying by 11:59 p.m. tonight your federal taxes. These are critical for funding the agencies that have been the backbone of the work that I will tell you about today, the National Institutes of Health and the National Science Foundation. But I underscore that the federal government provides the backbone of this work, and it's been supported by a variety of institutes and state agencies as well. Thanks to all of those. But more importantly, thanks to all the students, postdocs, undergrads, technicians, and other folks who've been in the lab directly and who've been critical to this work, along with great core facility directors and collaborators. I'm going to call them out as we go along, but I really always want to start off talks with thanking the people who've done by far and away the vast bulk of the work that you're going to see in front of you. So fundamentally, the lab is interested in understanding a question that I think motivates many people in the biological sciences: how cells and organisms know what's going on in the world around them. Our particular take on this has been to go through and ask broadly, how can conserved protein sensory domains be repeatedly used and used and used again in biology despite really different inputs and outputs? I show here, for example, 3 different biological systems: plants sensing blue light, cells inside of us sensing hypoxia, cells inside marine organisms sensing environmental pollutants. While these are radically different areas of biology and radically different things that trigger them, they're all united by the common use of small ligand-regulated protein-protein interaction domains known as the per-arc-sim or PAS domains. This, in my opinion, is the domain version of something we're real familiar with in other settings. Those of us over in eukaryotic signaling know G protein-coupled receptors real well as complete proteins that span the cell membrane to sense what's going on outside a cell and transmit it inside a cell. PAS domains get used, I think, even more broadly for a similar idea of sensing diverse inputs and controlling even more diverse outputs than GPCRs do. So when the lab opened in 1998, we knew only a little bit about them. We knew 3 atomic structures shown here, showing a mixed alpha-beta fold of about 120 amino acids. And we knew that these domains existed in some places that got us kind of interested in sensing and signaling. For example, we can see oxygen sensors built around heme, blue light sensors built around hydroxysuccinic acid. And as a formerly trained biochemist, the ones that get me most excited are the ones where we think there may be a trigger, but we're not lucky enough to find that as we're overexpressing pieces of proteins out of eukaryotic or out of bacterial expression systems, and we need to figure out what those triggers might actually be. So fundamentally, what the pass domains often do is bind other proteins, either in cis or in trans, and use the ligand state here to be able to control those interactions and in turn how they regulate those bound effectors. So what I really want to focus on is two exemplars of our work today, or two broader themes that have gone through the lab during our time. First is to ask pretty simply, some of the pass domains sense blue light by sensing a variety of flavin chromophores. We call these LOB domains, and they do very specific photochemistry in response to illumination. There are literally thousands of those LOB domains now, and they control many different diverse kinds of outputs. Serine threonine kinases, DNA binding domains, parts of the ubiquitination machinery, you name it. How can you use one common kind of input, one common kind of sensor to control many different kinds of biological activities? And number two, can we learn a bit from natural ligand regulation of some of these systems to therapeutically relevant artificial control? And we have some great examples of that out of the oxygen sensing system inside of all of us, built around pass domain containing transcription factors called the HIFs, the high hypoxia inducible factors. I think these two sets of stories will show the kinds of interdisciplinary work we do, rooted in structural biology and biochemistry, and also show our commitment to taking ideas in the lab and finding good ways to apply them more broadly. First question first, how can one kind of sensor control many different kinds of outputs? So schematically here, the pass domain subfamily we're talking about is shown in green, the LOB domains. They have a bound flavin chromophore like FMN, FAD, or riboflavin, but we find them from genomic sequencing attached to many different kinds of diverse effectors. Like I mentioned before, kinases, nucleic acid binding domains, you name it. So how can you use one kind of input domain to control many different kinds of outputs? A little bit of background here. There are, as of today, literally 8,000 plus proteins from bacteria, plants, and algae that contain these so-called LOB domains. They were originally found in a class of blue light regulated serine threonine kinases from plants known as the phototropins. The one that some of you on the call and some of you in the room may be familiar with is so-called AS-LOB2. This is a shorthand for Avena sativa phototropin 1 second LOB domain. This is where this all comes from. It's a blue light sensor in oat. And what we see from atomic structures, so in my lab by NMR spectroscopy and several other groups by X-ray crystallography, is the same kind of alpha-beta fold I showed you on the initial slide. A bound flavin chromophores in the middle, and some very elegant photochemistry from a variety of different spectroscopic groups showed what happens is in the dark state, you have a non-covalently bound flavin in an oxidized form. Blue light illumination drives radical pair photochemistry that results in the reduction of the flavin and concomitant attack of a cystenyl sulfur into the C4A position here. So dark state, non-covalently bound oxidized flavin, lit state, covalently bound reduced flavin. And that happens quite efficiently in many of the LOB domains. When you shut the light off, you spontaneously revert now back into the dark state. Question is, as a structural biologist, how do you relay that information out to control a kinase, a DNA binding domain, you name it? And we used a variety of technologies, including solution NMR spectroscopy, to be able to study not only the dark state and lit state of this AS-LOB2 domain, but how they interconvert in real time. In summarizing that work, what we see in the dark state is that alpha-beta fold with the, again, non-covalently bound oxidized flavin. And then at the C-terminal end is a auxiliary piece. It's not part of this conserved pass fold, but is conserved amongst the different phototropins of an amphithetic alpha helix. Yellow show various proton-proton distances that allowed us to unambiguously dock where it sits on the surface of the protein in the dark state. When you illuminate, you go through that photochemistry I mentioned before, and you cause the alpha-beta fold to become somewhat distorted. However, where this really has a big effect is the so-called J-alpha helix there. That auxiliary helix now completely unfolds and detaches off the surface of the domain. And that is an integral part of kinase activation as we can test, for example, by peppering the alpha helix with point mutations to forbid it from ever docking onto the helix or onto the beta sheet surface here. When you do so, you get constitutive kinase activity. So NMR, amongst other solution and biophysical and structural methods, helps show that what we've got here is a light-dependent protein folding switch. Whatever's going on at the chromophore affects whatever's going on at the alpha helix, and that in turn is integral to regulating kinase activity. Again, we looked at that by point mutations in the full-length protein. Mechanism was nicely borne out computationally in molecular dynamics work that we did together with the late Klaus Schulten at the University of Illinois at Champaign-Urbana. And really critically, I think one area on the application front that's been wonderful to watch is to see how people have started very creatively harnessing this helix on, helix off equilibrium into a wide variety of artificial settings. One of the earliest of these was a nice collaboration between Brian Kuhlman and Klaus Hahn at the University of North Carolina, where they just fused ASLOV2 onto one terminus of the small GTPase Rac1 and managed to get photo-dependent activity of LOV2 to be able to control the catalytic activity of the GTPase Rac1. More commonly, what we find in the current day and age, here represented by some collaborative work between my group and Kim Reynolds at Johns Hopkins University, is for ones where people go through, such as Kim and her group did into design, to take a perfectly good enzyme, say like DHFR, find a loop, a surface loop, splice ASLOV2 into this, and use that to find creative ways to transmit this helix on-off equilibrium to control activity, again, back at the active site. But the question I've got, I'm, with all due respect, I'm a scientist far more than I'm an engineer. This is what you can do in the lab, but is this what other natural proteins like the phototropins and other natural proteins use? Like I mentioned, our point mutation work over at phototropin clearly indicated yes. We've got additional data on the photes that I can talk about another time, but I want to focus on showing how this is even more general than just a kinase regulatory mechanism. So in this work, which really started in my lab when we were located at UT Southwestern Medical Center in Dallas, three very talented women in the lab, Abby Nash, an MD PhD student, Hiyamara Vera-Cancel and Laura Motomena, two postdocs in the lab, tackled this. Abby took on solution biophysics of this bacterial LOV helix turn helix DNA binding domain fusion, natural protein called EL222. Taking a look at the two kinds of domains, the regulation seems pretty clear. This is going to be a light-dependent DNA binder is the hypothesis. To test that, Abby used NMR. We were joined by Hartmut Luecke and Reggie McNulty, then at UC Irvine, for an X-ray structure of the dark state. And our integrated work between the solution NMR and X-ray work showed that what we have in the dark is something now, again, with the photosensory domain up top. We now have the connecting helix at the immediate C terminus here, the so-called J alpha helix. In this case, we think it's serving a passive role, but really intriguingly, the 4 alpha helix here adopts a position on the beta sheet surface, highly analogous to what we'd seen for ASLOV2 with the J alpha. Again, I cannot underscore strong enough that beta sheet surface is sensing what's going on for the illumination state at the chromophore, consistent with our model that this could be a light-activated transcription factor. In the dark, it's inactive because this helix, which I need to underscore is part of a dimerization surface essential for HTH binding, is now hidden up against the beta sheet surface in an occluded format. Two of my CUNY graduate students, Uthama Edipukani and Matt Clear, took on the challenge of trying to get structural information on the lit state here, which is a fair bit more complicated because each of the 3 primary structural biology methods, NMR, X-ray, and cryo-EM, all have problems in characterizing the dynamic lit state of this system. Have our combination of methods, hydrogen deuterium exchange by mass spectrometry, and EPR spectroscopy using the so-called DEER experiment to measure distances between spin labels. Let us go take a look at this protein not only in the dark state, but in the lit state on its own, and in the lit state in the presence of high affinity DNAs that we'd been able to get through biochemical studies. Integrating their structural work together using Alexander Bonvin's Haddock modeling package, we end up then being able to see now what light does. It changes the chromophore, as we've got in all the LOV domains, But now that's used to relay release of that so-called 4-alpha helix, converting the dark state inactive monomer into a lit state active dimer, where LOV-LOV and HTH-HTH interactions are essential for building an active transcription factor. What we've done with this now from a basic biochemistry standpoint is turn it into a tool that many labs can use. So our fundamental biochemical understanding of structural activation let us go through now and develop cases where we can not only photoactivate it in an NMR, such as we have here in the top left, but we can actually go through and get it to work in cells, not only bacterial cells as its native host, but convert it over into use for eukaryotic systems. And this has had wonderful applications from biotechnology, where people can go precisely control illumination and gene transcription inside of cells and culture simply by changing illumination protocols in real time, over to developmental biology, where work together with Anna Reed and coworkers at UC Merced have turned this into a powerful tool to go study zebrafish development, where you can go through and turn on genes at specific times in specific parts of developing embryos to turn on anything from fluorescent reporters to cell-specific toxins or signaling molecules to examine the impact of turning those on and off at specific times in development. More broadly, I really want to underscore for people that LOV domains are a subset of PAS domains. So the LOV domains like ASLOV2, shown here initially, and EL222 are using flavins to sense light. And they're doing so by harnessing these configurational changes at the chromophore and using them to affect the occupancy of this so-called J-alpha helix or 4-alpha helix, depending on which system we're looking at, as how it's bound up against the beta sheet. And I cannot underscore strongly enough, that's happening here for a helix that's immediately after the LOV domain. Here it's happening with a helix about 80 amino acids, C-terminal of the LOV domain. Well, if you keep looking even more broadly, we can see that the PAS domains have picked up on this trick too. And this is work by Jimmy Ciclari, a CUNY PhD student who's just wrapped up in my lab, now a postdoc with Ann Stock at Rutgers. And here what Jimmy was able to take a look at is something with the same kind of domain architecture, a PAS domain and a DNA binding domain. But in this case, it turns out that it's binding heme. So we think that we've got a redox or gas sensor on our hands, and it's doing so by binding onto the same beta sheet surface a helix that is regulated in its occupancy on-off, either by redox or by gas or by small molecule binding into the heme. And this piece of the helix that's grabbing is an N-terminal helix. So these PAS domains are looking here like very general ligand regulatable switches that we might be able to take advantage of. Nature's taken advantage of for millions of years to sense a diverse set of stimuli. So to summarize the work, how does a single kind of sensor control different outputs? Answers is we've seen a common use of so-called effector release. Hold on to a protein-protein interaction in one state, release it in another. That's been used by many LOV and PAS domains to develop novel not only great understandings of fundamental biochemistry, but develop understandings of novel mechanism-based biotech reagents and approaches. I should note there are other signaling systems we've seen in some other parts of LOV domains or in PAS domains, so there's still work ahead for us, but it's clear that this is a commonly used mechanism that we've been able to exploit for a wide variety of purposes, as have many people in the community. Let's switch gears a bit. Let's make this a little bit tougher. And now, how do you control PAS domains now by a diffusible ligand, and something that isn't binding, tightly and has a nice color to it, but something where we might be able to control protein-protein interactions by supply or release of an exogenous small molecule that we might be able to provide. This gets us over to the hypoxia-inducible factors, which involve some elegant natural regulation, as highlighted in the 2019 Nobel Prize in Physiology or Medicine to Greg Semenza, Peter Radcliffe, and Bill Kalin. Greg, Peter, and Bill's work can be summarized briefly here, that when O2 levels fall inside of us and other higher eukaryotes, you want to build a transcription factor built out of a heterodimer of an ARNT subunit that's oxygen independent and an O2 dependent HIF subunit, when the HIF alphas. So let's focus on the HIF alpha. That's where the O2 sensing comes in. In this case, the alphas are always being made, and if O2 levels are normal, you're going to go through oxygen-dependent post-translational modifications over in this arrow schematically represented to the right. These do a variety of things to downregulate HIF abundance and activity. The one I want to highlight in particular is a hydroxyproline residue that's specifically generated under normoxic conditions, recruits an E3 ubiquitin ligase, PVHL. That leads to polyubiquitination of the alpha and leads to its destruction. When O2 levels fall, taking O2 below the Km of the modifying enzymes, alpha accumulates, enters the nucleus, finds ARNT, where it controls a transcriptional response program of several hundred genes, which do all the things that one might imagine a cell starved for O2 would want to do. Increase oxygen carrying capacity of the blood by producing erythropoietin. Go generate potent angiogenic factors to recruit blood vessels into the O2 starved area, upregulate many of the enzymes involved in the glycolytic machinery, help generate ATP even when you don't have O2 as a terminal electron acceptor. That's great when you transiently turn these systems on and off in response to a variety of stimuli. However, if they're constitutively turned on, you end up predisposing cells very strongly towards various cancers, especially as nicely shown by the overexpression of HIF2 being highly linked to development of kidney cancers and a variety of other tumors. That's nicely exemplified by the von Hippel-Lindau syndrome, where the ubiquitination machinery that I noted up at the top is no longer working properly. That led several of us, my lab really tightly working with Rick Bruick when we were both colleagues at UT Southwestern Medical Center, to ask what if we could target those cancers not by going after the individual end products, don't go after VEGF or enzymes in the glycolytic machinery, but what if we could go to the regulator itself, taking advantage of the fact that there are pass domains present in the HIF alphas and ARNT. Would there be opportunities for us to be able to go in and find a small molecule way to control these, vaguely analogous to anything like you saw in the first chapter, as nature uses, for example, by sensing light through the law of domains, or in Jimmy Siclari's new work, O2 gas or redox state. This is a challenging thing to throw down. I cannot underscore that strongly enough within the time that we have to work together today. Protein-protein interactions are notoriously challenging targets for small molecule intervention. We hoped that the pass domains would give us a leg into that by having some natural vestige of being able to be regulated by small molecule ligands. To test that, we used a traditional so-called divide and conquer approach, expressing different individual domains of HIF, several HIF alphas and of ARNT. This is work really spearheaded by early Dallas members of the group, Carlos Amezcua, Paul Erbel, Paul Card, and Tom Sherman. And they were very successful in not only getting NMR and X-ray approaches online to be able to study this. So we have chemical shifts, we have solution and crystal structures, we can do small molecule screening by a variety of methods, including NMR spectroscopy. And that showed us a shocking result. Not only did we find small molecules that could bind into the core of human HIF2 alpha pass B, but a crystal structure really nicely done by Tom Sherman and Jason Key, postdocs in the lab at the time, showed that the center of the HIF2 alpha pass B domain contains an internal cavity of about 290 cubic angstroms in volume, with 8 crystallographically ordered water molecules inside, which have a beautiful hydrogen bonding network between each other and specific side chains surrounding the pocket. So we have here a big bag of water living inside of a protein domain. Any structural biologist or biochemist on the call knows this is very unusual. This is not the traditional hydrophobic core. Most proteins that show water-filled cavities of this size are usually apoforms of ligand-binding proteins. And from the NMR, we already had small molecules that would be binding into this region. And this is where I cannot underscore strongly enough, running a research lab with some very talented people in it who work in really different areas really glued a lot of this together. The first chapter that I showed you was all about the law of domains using photochemistry around a flavin bound up at the top. For our HIF story, the big bag of water is located in an exactly analogous location, despite the fact that these have nothing to do with each other biologically. They're not even in the same kinds of organisms. So could we then be able to turn what were small molecule binders into here into something efficacious? And this was a wonderful collaboration between my lab, Rick Bruick's lab. I need to give a shout out to additional collaborators who joined in on the chemistry side, specifically Uttam Tambar and John McMillan. Also really need to give really high marks to Bruce Posner and colleagues at UT Southwestern's high throughput screening facility that allowed us to go through and take what were initial NMR hits that we had already quite potent sub-micromolar, despite only looking at small molecule fragments about 200 molecular weight. These all go bind into the cavity that I just showed you. Those also gave us both raw materials and motivation to conduct a conventional high throughput screen looking at disruption of a protein-protein complex. It got us even more elaborate compounds and got us more potent, such that these compounds now were able to disrupt HIF activity for full-length proteins in living cells, disrupting transcriptional regulation off of those. At that point, as we were beginning xenograft work and taking a look for in vivo efficacy, we clearly began hitting a limit as to what we could do, even within the context of a top-tier US biomedical research facility at UT Southwestern. So Rick and I joined forces with several other people, helped with the formation of a small molecule drug discovery company called Peloton Therapeutics. Here I need to give a lot of credit to John Josie, Eli Wallace, and others who helped get this set up, who then continued the prosecution of this campaign, made compounds not only a little more potent, but far more suitable for use in animal and human model systems, leading then to Peloton developing not one, but two small molecule therapeutics that have gone into the clinic. The second of these, PT2977, is now fully in the clinic after Merck's 2019 acquisition of Peloton. Is used clinically now as belzutifan. And it ended up going through expedited review and deployment into the clinic based on extremely strong phase 2 data, showing, for example, single treatment, once a day treatment of belzutifan in a phase 2 for 61 patients who had that VHL disease, kidney cancers due to misregulation of HIF2. Amazing results with substantial decrease in tumor burden, and critically then for quality of life, decrease in the number of times that they needed to go through and take advantage of what were current standard of care, which is surgical resection of portions of their kidney. So I cannot underscore strongly enough, we got to this point by a divide and conquer approach of isolated PAS-B domains, using those for structural biology, biophysics, and biochemistry. However, sometimes divide and conquer doesn't work out super well. In this case, a lot of our crystal structures on isolated PAS-B domains, where we'd stabilized a protein-protein interface of the single domains we saw in vitro, turned out to not reflect larger biological conditions where, as you went through, I've got to acknowledge some very nice crystallography work by Freydun Rastinajad and coworkers then at Sanford Burnham, where they were able to get larger HIF complexes to be able to crystallize. In this case, we could see that the protein-protein interface we thought we saw in the isolated domains turns out not to hold when you put other domains and other structural contacts around them. However, even the crystallography work, I think, has left some things unexplained, and that's work we're currently publishing in press at PNAS, where we've now switched off of a crystallographic platform shown by Rastinajad and coworkers over to cryo-EM platform, here with work really nicely spearheaded by a current PhD student in the lab, Joey Clausen, former PhD student, Jim Su, former postdoc, Meiling Zhang, where we've been able to go through and not only get cryo-EM structures of the small short DNAs of about 20 base pairs that had been used for the crystallographic studies. And here our cryo-EM studies and the crystallographic work are quite similar to each other, with one conspicuous note that we're seeing a fair amount of what appears to be domain mobility inside of these heterodimers of HIF alpha, HIF2 alpha in this case, and ARNT. The ARNT subunit appears to be quite flexible in these cases, which then got us to think a little bit about some 30-year-old data out of Greg Semenz's group in particular, showing that this 20 base pair DNA, which includes the hypoxia response element, while important, was not necessarily the whole story in hypoxia-driven promoters. In particular, Greg had really identified that longer DNAs, including not just the HRE shown here on the left, but auxiliary sequences were important for full transcriptional activation in a variety of different platforms, taking advantage of those data and now shifting over to 50 base pair DNAs that are well set for cryo-EM shows that we get not only the structure that we saw previously by X-ray crystallography on the short pieces, but we actually see now a dimer of these heterodimers where a copy of two of these end up docking onto each other with binding across all the sites that were shown to be functionally important previously. That's really been nicely borne out then, taking advantage of that very flexible interface that we saw in the heterodimer cryo-EM. That ends up quieting down nicely as we build this so-called dimer of heterodimers, giving us some insights into protein-protein interactions that we think are absolutely critical here. So, and it nicely explains data not only from Greg's lab initially, but then more recently, Rosel Garcia and coworkers have a really elegant 2023 paper where they took a look at this and showed not only are both the HRE and the associated sequence critical for full gene activation, but actually there's quite a tight dependence on the spacing between the two, which is very important for in ways that we would have predicted from the cryo-EM. I want to just close where we're continuing to work in the areas by asking how, for example, dynamics are critical, for example, to get ligands in and out of HIF2 alpha PASB. That cavity is completely internal. Really nice work between my group and Lillian Chong's group at Pitt have given us both computation from Lillian's group and experimental work from my group here in New York, taking a look at entry and exit pathways and experimentally testing those as we go forward. In the interest of time, I want to sum up with what we discovered here. By following some of the lead that natural work in very non-biomedically critical settings over at plant and bacterial photosensing showed a lot of unexpected parallels where artificial small molecules can bind into the HIF2 alpha PASB cavity and control it in ways we think that are analogous to what the natural regulators use. This has led to a clinically deployed targeted cancer therapeutic of Merck's called belzutifan is being used now as frontline therapy for people with von Hippel-Lindau disease and as a second-line treatment for advanced renal cell carcinomas, which is an even larger group of people. And it's critical that you be able to go through and use NMR, X-ray, and cryo-EM to understand how these systems really work. I'll note that we and others have been looking to extend this idea to go regulate other PAS-containing transcription factors, including the clock BMAL system that's part of the circadian rhythm machinery. We have small molecules in joint work between my group and Carrie Parch's lab at UC Santa Cruz, just dropped in bioRxiv a couple days ago, and nice work by the aforementioned Rastinajad lab, published late last year in Nature Chemical Biology. Let me just take a minute for three big picture conclusions. Number one, no single technique got us to the answers we wanted. And I would say in 2026, I think it's time to retire some past conversations of my technique is better than yours. Folks, I just want to get to the answer and figure out how biology works normally and how we might be able to intervene with it artificially. And that has required here and required elsewhere X-ray crystallography, solution NMR spectroscopy, and cryo-EM. In order to do things like this, you need to build a proper toolbox of high-end instrumentation and, even more critically, really good people. This is my city block with my piece of CUNY here in the building in the left, part of another part of CUNY, the City College of New York, which I'm also part of in the middle, in New York Structural Biology Center, shown here on the right, which is CUNY and 8 other institutions. This group collectively has a collection of cryo-EM, NMR, and X-ray expertise that I think is unrivaled. And that happens because institutions, regions, and the nation weigh in together to make sure not only do we have really nice instrumentation, but the right people to be able to get good discoveries out of this. Another point I want to note is I'm getting a little tired of hearing that reductionism is dead. There was a very elegant speaker by at the ASBMB national meeting who made a specific point of this. In the interest of being gracious, I won't point out who that is, but I'll reply back that this is a picture of Toronto, Canada, one of my favorite places to be if I'm not in New York. And this is my view of what reductionism is. I'm showing you here a beautiful July afternoon, nice and sunny in Toronto, with the CN Tower being shown poking its head above the clouds. That's what we've got by reductionism, is a high resolution piece of understanding the bigger picture. But we're scientists. We understand the other forces around it. We can guess what's underneath in the clouds, and we can act on that and use that to drive biology forward. And finally, communication is absolutely essential between stakeholders. I started by thanking everyone for paying their federal taxes. These two things I'm showing here are united not just by the fact that they're large, complex, domed objects, but they're actually things that I think are really symbolic of what's needed here. We in the sciences understand what research looks like, but the people who are some of the most important stakeholders, the people who help set our budgets and more broadly, and the people who we have to thank on a regular basis don't always understand who we are, what we do, or why it's important. Everyone share a little bit of your science, even in a little way. Please, that is a really good thing for all of us. With that, I'm going to stop here. Thank everyone for their attention. And I'm more than glad to take questions relayed from Isabel and Kalsum. Thank you.
Thank you, Kevin. This was a fantastic presentation, super insightful. I really appreciate your time. We have 8 to 10 minutes to ask and answer questions. We have one in the Q&A. For Kevin, from the combinations look promising part of the drug development portion of your presentation, can you further explain if it's a combination of the current drug with others to tackle other cancer or tumors, or what exactly?
Right. So that is work that is not occurring within the context of my own laboratory. So I'm relying on public information from Merck and other companies that are working in this space. And yes, these are phase 3 clinical trials that are currently underway, looking at the combination of belzutifan and other small molecule therapeutics, looking for potential synergy such that you get either a better percentage of people responding to chemotherapy or into different indications. With that, I think it's best that I just note, again, that's where I'm a doctor, but I'm not an oncologist. And I would encourage the question asker and other curious individuals to take a look at publicly available information that's available from companies conducting those trials.
Thank you.
Thank you.
I don't know if you have other questions on the live audience on your end.
Are there other questions here in the audience? We can do those too. I've got some head shakes. I've got one or two embarrassed people. Mr. Clausen's asking why I didn't show more figures from his beautiful paper. Coming soon at a non ASBMB journal, but next one will be at an ASBMB journal.
We appreciate that. We have another general question. Do you think AlphaFold prediction is as good as X-ray?
For looking at ground state structures, I am truly impressed with what AlphaFold and comparable approaches can do. I think that predicting protein ligand complexes are still a work in progress. I'm impressed with a variety of tools that seem to be getting developed every day. But I'll also note at the end of the day, I view those as a good place to develop a hypothesis and not an answer to the question at the end of the day. Have those guided our thinking as to what we might try studying? Absolutely. But am I going to stand here and say that's the proof? Especially if I'm going to ask other labs to then take what we've got and use it as a tool in their own group, or start talking to people about clinical application. I think both of those warrant an experimental validation that is at an equivalent, if not equivalent tier to the places they're looking to use it.
Mm-hmm. I have one more question and a shout out. Great and inspiring work.
Thank you.
Does HIF2 cavity play an intrinsic role units biology, perhaps by binding metabolites?
Yeah, so wonderful question, anonymous attendee. We have, as a result of our structural and biochemical work, we've generated a variety of HIF2 variants where the cavity is partially filled, such that small molecules can't get in. And so there's a nice PNAS paper from, I believe, 2014 or 2015 out of Rick Bruick's group, where they made a knock-in mouse where they filled the cavity and replaced the wild type HIF2, and you can see a variety of interesting physiological changes that result. I do firmly believe that nature has not evolved that cavity to just simply carry water around or provide a drug discovery target. And I think it does bind metabolites. I'll note, as one can guess, if there's a HIF2 alpha, there's a HIF1 alpha and a HIF3 alpha, and they have cavities of different sizes. And I think there's a variety of nicely published work showing, especially HIF3 alpha, which has the largest of the cavities, does bind some interesting compounds that metabolites that look to have O2 dependent steps either in their biosynthesis or degradation. So yeah, that's my guess, is that, and I think that poses some wonderful philosophical questions. The Nobel Prize goes to not just one, but actually two separate O2 dependent steps to control HIF abundance and activity. Why is there a third? Yeah, nature's got a lot of secrets left for us to work on.
Great. Kevin, any last takeaways, message for our audience?
Yeah, I'm going to go back to this slide. I'm pretty serious about this. I mean, people at my barber shop are tired of hearing about HIF and blue light. My dad is older and in a retirement community. His buddies at the Saturday morning coffee hour are getting their share of this. People on Capitol Hill, as part of my work with ASBMB's PAC committee, are hearing it too. I think that's really important. And, I think this has been, the past 14 months, one of the most constructive things that I think one can share about it is, I think when communication breaks down, people don't understand each other. And we get left in situations such as we're in now, where I think some people who are setting budgets don't appreciate what NIH and NSF can do. I cannot underscore strongly enough, there are other people who think that you go from an idea to a drug overnight, and that's all we do in this business. No, it's something different. And everyone on this call, whether you're a student, you're a PI, you're a technician, you're in industry, you're in academia, you have your own view as to what biomedical research looks like. Talk to your neighbors about it, talk to your family about it, talk to your friends about it. And the first words out of your mouth should include some version of thank you, because we're all in this, and U.S. biomedical research and discovery is some of the best in the world because of joint investments we've all made through the federal government for 70 years at NIH and NSF. And I hope this provides a couple of examples where I've seen that firsthand.
Great. Kevin, thank you so much for your time and expertise today. Thank you for sharing all of your research with us. And thanks, everybody. We hope to see you next month at ASBMB Breakthrough Webinars, and have a great rest of your day.
Thank you, everybody. Take care.